So I was talking about polymicrobials and their role in intestine and in health, in cancer, in multiple sclerosis and IBS, many other diseases. And actually, the most important part is that we do know a lot about microbials and polymicrobials, and not just from now, but from 1648, as Leeuwenhoek had looked between his teeth, looked under microscope and saw that there are a lot of bacteria there. And he was so delighted, he saw that we are not alone in this world. And everybody saw it either, and all kings bought microscopes and looked what is happening there. 200 years gone, but nothing happened. For a simple reason, bacteria were everywhere. If you left your soup on the table, there were bacteria there. If you had a wound, there were bacteria there. So everybody thought that bacteria are emerging from alone, so we can't do anything. And even Lister wrote 1848 that the surgeon had to have a specific smell. He didn't call it bacteria, but it was actually anaerobes, if we can recollect it now. And the surgeon, if he lost his knife on the earth, he took it and started to cut further for a simple reason. If there are no bacteria there, there will be bacteria there. So why should I do anything? So it was first Pasteur who showed that bacteria are not all made here, that they have and need specific conditions, and that we can investigate one bacterium by one, showing what can they do, how can they harm, and what can they do. And everything else was gone. So from that time point, the infection disease started, and everybody knew, okay, the pathogen should be in healthy, it should not be in diseased, and if you transfer it, it should cause a disease. So that was a paradigm, and it changed a lot. We have eradicated all infections, which we thought to be infections. But it's just one point of the story, since all infections that we do know using Pasteur method are monoinfections. It's just one infection. But how many polymicrobial infections do we know? Actually a lot. But what do we know about them? We can say appendicitis is polymicrobial. What we do? We make surgery. It's IBD, it's Crohn's disease, it's bacterial vaginosis, and probably the most of autoimmune diseases are likewise polymicrobial diseases, where a community is important, where some kind of band of bacteria which are cooperating and which are working and behaving absolutely different as a single bacterium in a single culture. So investigating them would be important to see what it happens, since until now we are just living in 1864. We made no progress still there. We are just by monoinfections. But the problem is, how can we make a progress just knowing what is there? What is there is not actually important. It's important where it is. So, for example, Neisseria meningitis is a germ which causes meningitis. Meningitis is a very severe disease. But actually, is it normal for your mouse? In 30% of healthy people, it's living there. So, is it pathogenic? Surely. But not if it is in your mouse, if it is in your meningia. So, it is quite important to show where bacteria are, what they are doing, how they are cooperating with each other. But in a moment, if we look what all are saying about is just there are a lot, and we are just trying to correlate what we find in composition Änderung with some diseases. Are they good? Are they bad? I don't think that it is a good way to get to decision about real pathogenic role of this bacteria. We have to show where they are, what they are doing, with what they are cooperating, and not just sequencing and saying, yes, they are there or they are not. We do know that there are a lot. And thus, the most presentation are saying, we have investigated that, we have investigated thus, and we have meto, nano, micro, and so on. And at the end, so what? We will investigate it further. But not coming to the end. Actually, what do we want? And we want to find the pathogenesis. How bacteria, groups of bacteria are cooperating with each other and what they are doing. Until we do it, until we care about problems of our patients and problems of our health, and not just making basic science, there is something there, we will have no progress. The locations are actually, you use molecular genetic methods to say where it is, but you take a sample, which is a biopsy, so it is intracellular, it is intramucous, it is on the cell, it is outside of cell, so you actually don't know what you are investigating. So without looking what is the functional anatomy, where is the bacterium or specific bug at specific point, if you have no histopathology of it, you don't understand what is happening. There is a Neisseria in your mouth, so is it bad, is it good? It's nothing. But if it is in meningia, it is something. There is Fusobacterium nucleatum in your mouth. Is it something? No, it is absolutely normal there. But if it has a specific histopathology in your appendix, it's very important. As long as we just take probes or samples based on microscopic view, a sample with a biopsy, a cut of histology, and making a very specific analysis with sequencing, what do we know? There is something somewhere there, and everything else is just a noise of names. Mucosa associated, it's a biopsy, it's not mucosa associated, mucus associated, fecal, and so on. What I started my talk with is about asking the doctors what is actually the colon for, and the most didn't know what for. So you know, the heart is for bringing blood further. Your muscle is for moving your body. Your head is for moving your thoughts, your kidney for removing your gifts from the body, but what is the colon for? What is the positive of it? So what we know, it is a reason for cancer, for IBD, for Crohn's disease, colitis, for diarrhea, for constipation, for ileus. But what is the positive of it? What can we call positive of it? For heart, we can say it makes blood, it has pressure, and so on. For thoughts, we can say there's some kind of physiology. But what can we say for colon? Stool frequency, stool smell, stool form? Is it really was something positive? So nobody knew it, but actually the answer is quite clear. It is a high sophisticated organ which we are not still grown to understand to. You see, the future of civilization is bound on biotechnology. We are getting gas, we are getting biological, which are very, very precious. We are getting insulin, all with molecular genetic methods. And the most important tool of it, it's a bioreactor. So bioreactor is where we are getting all this mass of substances to go. And I am sure that in the future we will get oil with it and everything else. It is just a question of time. But the evolution developed a bioreactor much earlier than we started our experiments with molecular genetics, and it is much more efficient. If you look at efficiency of our bioreactor, we can reach concentrations of 10 to the 10 for short periods of time. The colon reaches concentrations of 10 to the 30 for years. Our bioreactor, which we are working for producing of biologicals, are working with one bacterium. If we get 3 bacteria, we never know what happens in 2 hours. Colon is working with at least 5,000 different bacterial groups and other microorganisms, and is stable over years if it is working correctly. So the question is not what is in it, so how can we look how it works, and what it does. And if we look not at just in composition, but at criteria, it's the most interesting and important criterion, for example, high concentration. Each kind of species has bacterial groups which have to be there. Probably they are important for biofermentation. For human beings, there are just 3 groups. All other bacteria are individual, so we can steer over this bacteria the biological input of our bioreactor, and the benefits are enormous. So what we have to look is how these aims are reached, is the concentration is there, how they are distributed over a colon reactor, how stable they are over the time, how can they be influenced, and how can we be protected against it. Bioreactor is similar to a nucleic reactor. Everything is good as long as it is there and it is well protected. But everything is bad as soon as it comes out, like Fukushima. And if it comes out, nothing is good. And if it comes out, it makes cancer. So you can start arguing what is before. Cancer, bacteria, what is primary, what is secondary. But let us take an example of Fukushima. You have a tsunami, it's the first. You have then failures of administrations, failures of management, which takes days before they start with chilling of reactor. And then you have an outburst, you have a lot of sequences which are, and many people will die on cancer just from that. So nucleotides are not primary, surely. But is tsunami a reason for cancer? Surely not. Is a failure of administration reason for cancer? Surely not. Nucleotides, and each of them is quite specific, has quite specific effect on cells, on organs, which causes specific cancer regions. So it's like a bioreactor. As soon as your protective mechanisms are perfect, as soon as no bacteria has a contact with your epithelial surfaces, everything goes good. But in case you take emulsifiers, for example, or you take detergents, on your mucous layer is disturbed, bacteria come in contact with your epithelial cells, process starts which are germ related. So they are cancerogenic, since bacteria, E. coli with cytolytic distending toxin and so on, they will be very manifold, not less manifold than carcinogenicity caused by nucleotides gone from Fukushima. We have to investigate each of them, and we should start not there is a bacterium there. We should start to follow the whole chain from tsunami to disturbance to getting out, and what can we do, how can we protect, and so on. It is the same. Just having an instrument, I can sequence. So if I sequence, I can find it is correlating or it is not correlating. Correlation doesn't mean much. We need to understand what is behind it. So in a moment, microbiome is something like astrology. You see, our ancient people looked at the sun, looked at the stars, and they found quite quickly that some pictures of stars are correlating with weather, with changes of water in Nile, with a lot of things. So if these correlations are so important for so massive things, they would be important also for your habits, they would be important for your life. So the astrology came. We do know that they have no importance for this. They are just markers of what is happening in the universe. But there are a lot of people which gave money just for describing how the stars are moving and what an impact they have on something else. That's not the question. We have to find out what is behind. So we should start moving from, I have a bacterium somewhere and it is something and it is correlating with something else, but how does this correlation comes to?